Okay, so in the last video, we talked about promoters and transcription initiation. So how does RNA polymerase know where to start transcribing a gene? And in that video, we talked about how promoter sequences are specific DNA sequences that position your RNA polymerase to initiate a defined location in the genome and start transcribing from a defined strand. And the key idea was that the template strand is what's used to make a copy of the coding strand. So one strand you're going to get basically an exact copy of, but with the but it's going to be made out of RNA, and it's going to have uracils wherever you saw thymines before. And because the template and coding strand are both held by RNA polymerase when it polymerizes, so it's like opens it up and then does the polymerization activity, only one strand of DNA can be transcribed at a time. And I fixed this in the slides in the previous one, but not in this one, so I will fix that again. But now that we know how to start transcribing, it's just as important to know how to stop. So what prevents RNA polymerase from just going on forever and transcribing, basically making an RNA copy almost of the entire genome? And that's what we're going to learn about this video. We're going to learn about transcription termination, so the process by which RNA polymerase learns to stop. And so, as before, we learned about the promoter, the sequences at the start, the 5' end of a gene, which is say, let's put an RNA polymerase here and transcribe. So what is going to provide the signals to stop at the end of the gene? So polymerase, we would think, gets here, and something tells it, hey, stop making this transcript, we've made enough. And so that's the process that we're going to talk about in this lecture. And we're going to primarily focus on prokaryotes. So as I mentioned in the previous video, basically the prokaryotes and eukaryotes are always very similar conceptually in terms of what is how this type of regulation process works. It's just that the eukaryotic versions are often more complex and more convoluted, and basically ends up being like a lot of names and words. I think if you can understand the prokaryotic version, then you ought to be able to understand any eukaryotic version that you encounter. So let's go through the prokaryotic version in good detail, make sure we really understand it. And then we're poised to understand eukaryotic systems going forward, whether in this class or in other classes. And in prokaryotes, there are two primary mechanisms that can be used to terminate transcriptions. And so this polymerase is going along here, it's making RNA, it's got this nice bubble, and then there's going to be some mechanism here by which whoop, the bubble is closed, the polymerase leaves, and the RNA is produced here, right? And for each of the two mechanisms that do this termination activity we see here, the way that termination is going to happen is something is going to disrupt RNA polymerase and its ability to continue interacting with the template strand and advancing forward. And this disengagement with the template is going to cause the RNA polymerase to basically fall off the DNA. That transcription bubble is going to close and the process will be terminated. So what are some mechanisms that can be used to do this? So one of them is a process called row-dependent termination. And the idea here is that your RNA polymerase is going through, it's transcribing, and eventually it goes through a stretch of DNA that encodes what is called a row binding site. And that is a stretch of DNA that, when transcribed into RNA, is going to provide a binding site for a protein called row to assemble onto that RNA. And what is row? Well, row is a helicase. And you'll recall from the earlier lectures that helicases are enzymes that use ATP to translocate on RNA or on nucleic acids and unwind strands from one another, right? And so when this row factor binds to the RNA as it comes out of the polymerase here, so we've made it through here, this row binding site's been produced, row factor is going to land on here, and row is going to start, its helicase activity is going to fire up, and it's going to start moving towards the 3' end, and it's going to start trying to unwind the RNA-DNA hybrid that is what is holding RNA polymerase to this transcription bubble and providing the ability to add nucleotides to this 3' end here. And so we transcribe through this region, we generate a binding site for Rho, Rho hops on, it starts helicasing its way up here, and it's going to eventually pull those strands apart. Okay, and once the DNA RNA hybrid has been unwound here, then RNA polymerase isn't going to be able to continue to polymerize, right? The whole thing is going to basically fall apart, and as before, the polymerase is going to get ejected from the strands, and the DNA is going to seal up, and the RNA eventually will fall off. Okay, and so in this case, we're using a helicase activity to get this job done. And I just want you to kind of double down on this. We've seen helicases before. Remember, these aren't enzymes that are going to break covalent bonds. They're not nucleases. They don't cleave anything. They're simply going to unwind or melt nucleic acid hybrids, and this should say DNA or RNA hybrids here. It's not just specific to DNA. I apologize. And so what this activity is doing is it's breaking hydrogen bonds between strands, but it's not cleaving the background. And so when you think about what's happening here, essentially in this termination mechanism, the Rho helicase is peeling off the RNA from the DNA to stop this process. So I found this diagram here to be kind of nice. You've got your RNA polymerase transcription bubble here. Rho is going to start assembling on this and work its way down, and eventually it's going to get to your bubble, and it's going to unwind the RNA DNA and displace everything. And so you really are using a helicase to peel off the RNA from the DNA. Now there are other classes of termination that are called Rho-independent termination, and so as their name would suggest, these are termination mechanisms that do not depend on the activity of the Rho helicase. And so how are these ones going to work? Well, these are a particularly fun one to think about because they make use of actually many of the concepts that you've already learned about in terms of nucleic acid structure from the first couple lectures of the course. And so I think when you see these, you'll really appreciate how what you've learned can help you understand what's going on here. And in Rho-independent termination, what happens is the RNA transcribes through a stretch of DNA that's going to produce a very specific outcome in terms of the RNA transcript that's produced here, okay? And one of those things is it moves through a GC-rich region here. So you see there's a lot of GCs here, there's a lot of GCs here. And remember that RNAs, even though they're single-stranded, they can adopt secondary structures on their own. And we talked about in the earlier lectures, a hairpin structure was an example of this. And so it turns out this GC-rich region that gets transcribed can adopt a hairpin structure. And so that hairpin structure is shown here. And if you're feeling extra studious, you can kind of draw out this primary sequence and then see if you can come up with the same hairpin structure yourself. But the bottom line is you've got this nice stem loop structure here that produces this secondary structure. And then interestingly, what's right downstream of the hairpin, so getting closer to the 3' end where polymerase is trying to extend, is a long stretch of U's. So we basically have a U-rich segment here, and that means that in this portion of the RNA, the interactions between the RNA and the template strand are held together primarily by UA base pairing interactions. And so this structure is interesting because it causes two things to happen. One is that the RNA hairpin structure itself can interact with RNA polymerase in such a way to cause a stall. So this hairpin structure basically gums up, it's like a wrench in the works of RNA polymerase that makes it harder for the polymerase to keep transcribing forward. So that's one part of what this structure does. And the other is that the U-rich segment, which is what's actively making base pairing interactions with the template strands, well, this is made up exclusively of UA base pairing. And remember, we talked about in the lecture on base pairing earlier that A and U, or A and T, either one, only interact using two hydrogen bonds compared to 3 hydrogen bonds for CG. And so the interaction here between the RNA and the template strand is about as weak an RNA DNA interaction as you could possibly get. So it's the bare minimum number of hydrogen bonds here. And so using some of the terminology we heard, we would think that this has a very low TM here. And so this combination of being stalled, that is unable to advance forward, and then what you're stuck on here is a very weak interaction. Because it's the interaction between template and RNA that's really holding polymerase and this bubble together, it's so weak now that the whole thing is basically going to fall apart, and your polymerase complex is going to disassemble. And so this is just a nice example of how CG, making 3 bonds, can form a really stable structure with itself, whereas these UAs with the template and the RNA are so weak that it causes the whole thing basically to fall apart, right? And so this is another type of termination mechanism that we see. Now, what about eukaryotes? Well, as I indicated in the initiation, there are 3 different types of RNA polymerases in eukaryotes, and again, just in the same way that each of them has its own complex initiation requirements that might be distinct for each type of polymerase, so too does each polymerase have its own sort of specific and oftentimes very complex termination requirements. And so I would say the main thing to take away is that eukaryotes are going to have similar mechanisms for saying when to stop compared to prokaryotes. They're just going to be more complicated and more convoluted. And so for the purposes of this course, I really want you to understand the prokaryotic systems, and then be able to take those ideas and kind of contextualize. If someone tells you about eukaryotic transcription termination, you'll say, oh yeah, there has to be a way to get the RNA polymerase to fall off, or yada, yada, yada. So that's the main thing I would say about eukaryotic transcription termination at this stage. And so let's just sum up what we learned about transcriptional termination. So just like initiation, RNA polymerase needs a signal to stop transcribing so that the RNA that we produce doesn't go on forever. And we talked about 2 interesting mechanisms that are distinct in prokaryotes for doing this termination process. We talked about the Rho-dependent mechanism, in which we make use of a helicase called Rho, which after the RNA transcribes through some stretch, a binding site for Rho is produced on the RNA. It can assemble there, and then that helicase activity can essentially unwind and peel the RNA off from the RNA polymerase transcription bubble to stop transcription. So that was one mechanism, and we learned about the Rho-independent mechanism, and this is a mechanism that relies on formation of a very particular RNA structure, this RNA hairpin that's very stable and is kind of like gums up the extension process, along with a very a stretch of U's that make the interaction between RNA polymerase and the template strand very weak, and that combination of stalled and weak interactions causes the whole thing to fall apart. So those are specific examples that I think are really straightforward to understand at the prokaryotic level. And as I said, termination in eukaryotes is similar conceptually, like we need signals to stop, but more complicated in terms of the execution. And so I think most of those mechanisms are really outside the scope of an introductory course like this. But if you guys are interested in learning more about them, let me know, and we can talk about some of those, or I can make a video about that if that's something you guys want to know more about. Okay, so to sum up what we learned about in the transcription series of lectures today, hopefully you've learned that the main thing you really need to know is that DNA sequences are transcribed into RNA by RNA polymerase. RNA polymerase uses one strand of DNA as a template to generate the RNA polynucleotide. The start of transcription is specified by promoter, which is bound by RNA polymerase. Prokaryotes have a single polymerase that they can kind of modify in different ways using those sigma factors, whereas eukaryotics have multiple RNA polymerases and everything's more complicated in those systems. And so we focused on the prokaryotes, where transcription is terminated by either row-dependent mechanisms using a helicase or these row-independent hairpin mechanisms. And in eukaryotes, both initiation and termination are just more complicated. It's just the way it is. And I really want you guys to get this idea of start signals and stop signals and the architecture of the gene here under, really try to lock this knowledge in, because in the next series of videos, we're going to talk about how using these start signals and stop signals and creating regulation around them, it's going to be able to make it so that the cell doesn't transcribe all its genes all at the same time, right? And that regulation of transcription, that is deciding what genes to make at what times and when, is going to enable cells to be different and to react to different environments and change their states. And so this is when we're really going to start to get into the essence of how the biochemical knowledge we're learning so far is really leading to cells being living objects that respond and change to their environment. And it's also going to help explain how a single cell or a single genome, like what we have in our bodies here, can be used to make all kinds of different cells, like heart cells and neuron cells and so on and so forth, by choosing which genes are transcribed. And so this is, I think, when the course is really going to pivot into a very interesting biological set of things that we can learn. But in order to do that, we really need to lock down our understanding of the basic control logic of any gene. So really make sure you know this well before we go into the next lecture. And so with that, I will stop the video and I'll see you in the next video.